Hi friends, so now I'm going to talk about gene interaction. So in my previous video we have talked about Mendelian inheritance, the monohybrid cross, dihybrid cross and their ratio. But in this video we are going to talk about the non-Mendelian inheritance, which means the inheritance which does not follow the Mendelian ratio, which is 9 is to 3 is to 1, 9 is to 3 is to 3 is to 1. So, but we are going to talk about different gene interaction and how they change the ratio of Mendelian inheritance. So, please make sure to like, share, and subscribe my channel. So, first, uh, before the Mendel and the other co-workers, they assumed that a character was governed by a single gene. That's why they had the monohybrid cross. But it was later discovered that in organisms, one character is mostly controlled by two or more genes, and they can affect the development of certain characters in different ways, which means that the one gene is affecting the expression of the other genes, or both the genes are responsible for developing one character. So this is what we are going to talk about in this video, and this gene can affect the character in different ways. So what is gene interaction is the phenomena of two or more genes, like two or more genes affecting the expression of each other, like we have talked about. Suppose you want to develop the flower color, which depends on the two genes. Like if one of the gene is not present, it cannot develop the flower color it wants, or like when only both of them are present, then only this color will be developed. So, if this is the example in which two or more genes affect the expression of each other to develop single character, right? So, this is called gene interaction, like the interaction between different genes. So, there are different types of gene interaction. The one is the intra-allelic or allelic gene interaction, which we have talked about is the case of incomplete and codominance. So in this case only one gene we are talking about, but the alleles they interact in a way to produce the new character. So if you remember, in the incomplete dominance in my previous video we have talked about, so neither of them is completely dominant on the recessive one, right? And the the offspring that we get is the intermediate of both dominant and recessive. So this is called the incomplete dominance. So the alleles are changing something to produce a color which is the intermediate of the two, neither of the parents. And the codominance, in which both the alleles are equally expressed and both are equally dominant. So this is like the intra-allelic, because this is controlled by one gene, but different alleles are regulating and producing the new character. And then there is another one which is called the non-allelic or inter-allelic. Like inter means between different alleles of different genes. So in intra-allelic interaction, the two genes we are talking about, two or more than two, they can be located on one chromosome or in a different chromosome, but they interact with each other for the expression of single phenotypic trait. Like like we have talked about, one two genes controlling the flower color. So the alleles are present on same chromosome or in different chromosome, but they interact with each other for the expression. And there are basically four different types of the gene non-allelic gene interaction, which we are going to talk about in this video. So the first one is the complementary gene interaction, in which two genes complement each other to produce a single phenotype, and we are going to talk about it in the later video. And the epistasis means the one gene is masking or hiding the expression of the another gene. So it can be dominant recessive or dominant epistasis, or the recessive epistasis means the dominant gene is hiding the expression of the recessive one, or the vice versa. And then duplicating factor, in which both the factors are required to, like either one of them is present, then also we get the same phenotype. And supplementary gene action means like you have uh, one gene present, it produces a particular phenotype. But when the another gene is present, the phenotype get supplementation and then it expresses the another phenotype. So first we are going to talk about is complementary gene action. So in this type, two or more genes like dominant genes which can be present on the same chromosome or with different chromosome, when interact with each other produce the character. But if the other one is not present, it will not produce. Like suppose there are two genes which are controlling the expression of some character like height. Suppose there are two genes controlling the character height, so only when both of them are present either in homozygous condition or in heterozygous condition, like the dominant one homozygous dominant or heterozygous dominant, so when both of them are present, then only you will see the phenotype. When either of them is not present, like one of them is not present, then you will not see that phenotype. So this is called the complementary, which means both of them are required to produce a particular phenotype. So instead of the 9 is to 3 is to 3 is to 1 ratio that we see in Mendelian inheritance, in complementary gene action, what we get is 9 is to 7 ratio. So I will show you one example. So here you can see we are talking about the flower. So this is, both are white. So here you can see it is expressed by the, like it is controlled by the two genes CC and PP. In both of these cases, it is white because in the first one C is dominant but the P is not present because it's in recessive condition. And in the parent 2, the C is in recessive, P is dominant. Still it's white because C is recessive. For this to be purple, we need both of them to be dominant either in homozygous or in heterozygous condition. So when we cross this flower, what we get is all purple because we get the heterozygous, like the capital C from here, capital P from here. And in the F1 generation, we have both the genes present in the heterozygous condition, heterozygous. So both the genes are dominant and what we get is purple color. So when we fertilize, like when there is self-fertilization between the F1 generation, what we get is either they need to be both in the homozygous dominant or in heterozygous condition to produce the purple flower. The rest of the case will be like the white flower. So here you can see when either of the alleles C or P is in dominant, both is in dominant condition, can be one allele or both the alleles, the phenotype that we get is purple. In rest of the case, like when C is dominant but P is in recessive, or the vice versa, like C is recessive, P is dominant, or both are recessive, we will get white phenotype, white color. So instead of the, like complementary means we require both the genes, both the alleles in dominant condition to produce the purple color. Otherwise what we get is white color. So this is complementary gene ratio and the ratio complementary gene action, and the ratio is 9 purple and 7 white flowers. So I hope this is clear. So next we are going to talk about is epistasis. So as I have mentioned in brief that an allelic pair or the gene, it suppresses the activity of the another gene. They can be in the same locus, right? So suppose we have the height, or like the, suppose we have the flower color that we have talked about. So in that case, one allelic pair, suppose the other one like B, it hides the expression of the A. So whenever B is present, it will hide the expression of the A, and what we get is the B color. So this phenomena is called the epistasis, and the gene whose expression is inhibited. So we have talked about like whenever B is present, it will hide A. So A is called hypostatic gene and B is called epistatic gene. So it has two types, dominant epistasis or recessive epistasis. So I will talk about both, what is dominant epistasis and what is recessive epistasis. So what is dominant epistasis is when the dominant allele prevents the expression of the another one. So suppose B is present in the dominant condition, and whenever the B is in dominant, it will hide the expression of the A. So when it will be recessive, it will have no effect on A. Only when the B is in dominant condition, it will hide the expression of A. That's why it is called dominant epistasis. And the ratio what we get is 12:3:1. So like suppose we are talking about here the coat color, like suppose we have the gene which is BB. So as I have talked about, the BB gene is epistatic and AA is hypostatic because we are hiding the expression of A. So capital B or like homozygous dominant or the heterozygous, because they are in dominant condition, this is white. So whenever this case is present, we will see white because A codes for yellow, but because B is hiding A, so whenever this is in dominant condition, what we get is white instead of yellow. So here you can see the examples. So when we cross this F1 generation, so whenever the B is in dominant condition, the color we get is white. Whenever B is in recessive condition, B now cannot hide the expression of the A. So what we get is the color of A, which is yellow. So here you can see in the white one, it doesn't matter whether A is in homozygous dominant, recessive or heterozygous. But because B is in dominant condition, we get the B color. Only when they both are in small, we get the green color. So I hope it's clear because it's dominant epistasis. And the ratio we get is 12 white, 3 yellow and 1 green. So now we are going to talk about recessive epistasis. It's the opposite of dominant. So whenever B is in recessive condition, it will hide A. Otherwise, we will see the A color. And the ratio we get is 9:3:4. So suppose we are talking about the coat color, which can be agouti or albino. So in this case, the small a is epistatic, means that the AA is hiding B. So whenever AA is present, what we get is albino. But whenever AA is in dominant condition, we get the phenotype of BB. So here you can see when we cross this parent, in this case A is in capital form, like in dominant phenotype, dominant allele. So now is because it's dominant, it cannot hide B. So what we get is the phenotype of B agouti. Only when, here you can see, only when AA is in recessive condition, we will see the albino, like the phenotype of recessive one, because now it's hiding B. So what we get is albino. So in overall, the ratio that you get is 9:3:4, which is different from the Mendelian one. So I hope the epistasis, dominant and recessive is clear. So another one is duplicating factor. So it is controlled by two genes, right? But if one is also present, that is enough to produce the phenotype. Like when both of them are present, you will still see the same phenotype as one of them. So there is no enhancement by both of them. So there is only one is also enough. So here you can see like the ratio we get is 15:1. So here you can see, suppose we are talking about some color, the orange and the white, like the brown or the white, whatever. Even like when both of them are present, you will get brown. When only one of them is present, like here you can see small a, but the B is capital, then also you will get the brown. But only when neither of them is present in the dominant condition, then only you will get white, right? So in this all of the scenario, what we get is brown color. Only when both of them are in recessive condition, we get white. So the ratio that we get is 15:1. So this is called duplicating. Like Duplicating genes are controlling the factor like the trait, and even one of them is enough. So next is the supplementary gene interaction, as I have explained in brief like before. So they interact in a way that one dominant gene produces its effect and the other one is present or not. But when the second one is present, a new character is produced. Like in duplication, what we get is when both of them are present, we will still getting the same phenotype. When single one is present, we were still getting the same phenotype. But in supplementary, what happens is we get the one phenotype with the one gene, but when the new, like the other gene is also dominant, the expression is changed, and the ratio that we get is 9 is to 3 is to 4, which is the same as the recessive epistasis. So here you can see the example. So suppose we are talking about the grain color, which is purple. So here you get the, like, because here it's all like small and small, so when these parents are crossed, like what we get is the purple F1 generation. So when we cross them, when both of them are present, we get purple. But when either of them are present, like when the red one, like the RR is present, we get the red color, and when the PP is present, we get white color. So whenever they are individually dominant, like either the R is dominant but P is recessive, and the P is dominant, R is recessive, we get different color like red or white. But when only they both are present, we get purple color. So this is purple 9 when both are present, red when only R is dominant, and white when only P is dominant. So this is called supplementary gene interaction, which means that the presence of another gene is supplementing the first one and we are getting a new phenotype. So I hope this gene interaction, non-Mendelian inheritance is clear. And if you have any doubts, please let me know in the comment section. And please like, share and subscribe my channel. Thank you.